This lecture is on the basics about the mycology laboratory. It will include features that will be helpful for both a review for your AP or CP exam. On the first slide, we're going to talk about the starting points in mycology. And there's two forms of organisms in mycology, yeast and molds. Yeast are unicellular. They produce budding daughter cells. And when you grow these organisms on your solid media, they look like bacterial colonies, white and beige and a little bit creamy. Some of the genera do produce drier colonies and some even produce mucoid colonies. On the next slide, we will talk about what molds basically look like. Molds are very different than the yeast organisms, of course. They are filamentous with hyphae, and they produce colonies on agar plates that look fuzzy or hairy. They produce conidia or spores, and the colonies on solid agar are either downy, fluffy, cottony, and molds can experience some beautiful colors that can be very helpful in their identification. On the next slide, we'll talk about appropriate specimens and transport conditions for fungal disease diagnosis. Fungi are very hardy organisms, and they can grow from many of the specimens that are sent to microbiology for culture. They don't require any special transport media. Sterile containers are quite adequate for the transport of specimens. You just have to make sure that you're eliminating bacterial contaminants, because bacteria grow faster than the yeast or molds and can overgrow these organisms and perhaps eliminate them from a culture. You can submit respiratory specimens, tissue biopsies, cutaneous biopsies and specimens, ocular, sterile body fluids, blood, and bone marrow. All of these are capable of growing fungal organisms. On the next slide, we start talking about the fungal culture media. The media that we use in the mycology laboratory is different than that used in bacteriology. Two of the most common media that you hear about in the mycology laboratory is the Sabouraud's glucose agar, or known as SABs, and inhibitory mold agar. Sabouraud's agar is really an all-purpose fungal media. The only problem with Sabouraud's is it does not discourage the growth of bacteria. So if you have a specimen that is loaded with both bacteria but some yeast, the bacteria could actually overgrow the yeast. So that's one of the bad things about Sabouraud's. Oftentimes this media is used more for subculturing and working up of organisms more than it is the initial isolation from patient specimens. It contains 2% glucose and the pH is around 7. The most commonly used fungal media these days is inhibitory mold agar. This is a selective SABs medium that contains chloramphenicol and gentamicin, two antibiotics that will help inhibit bacterial growth, and also some enrichment that helps nourish for the growth of both yeast and mold. It's also good for the primary recovery of your dimorphic pathogenic fungi, and saprophytic fungi and dermatophytes are somewhat inhibited on inhibitory mold agar, helping the more pathogenic fungi grow. On the next slide, we talk about more common fungal media, including mycocell and mycobiotic agars. This is a selective SABs formulation that contains chloramphenicol and cyclohexamide. These agars are primarily used for the culture of dermatophytes, which of course are infections that occur in skin, hair, and nails. Brain-heart infusion is also commonly used in mycology laboratories. It can recover all fungal organisms. It is made selective by adding chloramphenicol and cyclohexamide, and you can also add blood to this agar to nurture for the systemic fungi. Once you have your specimens inoculated onto one of these fungal medias, all fungal culture should be incubated for 4 weeks at 30 degrees centigrade. This is a lower temperature than you would normally think of for isolation of organisms. Bacterial isolation is done at 35 degrees centigrade, but the temperature of choice is 30 degrees centigrade for looking for molds and yeast. This helps nurture the mold form of the dimorphic fungi and overall works very well for the isolation of yeast and molds. On the next slide, we start talking about the effects of cyclohexamide when added to media. Cyclohexamide is found in a number of fungal media that we use, and it can help prevent rapidly growing molds from overgrowing the dimorphics and dermatophytes that grow a bit slower. So that's the good side of cyclohexamide. But cyclohexamide also has a bad side. You have to beware and be aware that it can inhibit the growth of very important organisms such as Trichosporon or Cryptococcus neoformans. On the next slide, we talk about the processing of fungal cultures. The stepwise processing includes, of course, the first step being to inoculate the fungal media that you're choosing to use on that patient specimen. The second step would be you need to seal the plates with tape or some kind of a shrink seal to prevent culture contamination and escape of fungal spores from the growing culture. All cultures are then put in an incubator at 30 degrees centigrade for 4 weeks. If growth occurs, the proper identification tests need to be done, depending on whether it is a yeast or a mold. Yeast identification methods are somewhat similar to bacterial methods. The most common methods used in mycology laboratories are manual and automated biochemical reactions, and they formulate a biochemical pattern that can be used to identify the yeast. There are newer methods, however, that has also been applied to yeast identification, and these include mass spectrometry, such as MALDI-TOF, and also 6S RNA sequencing that can also be used in more sophisticated mycology laboratories, particularly to identify some of the more difficult to identify yeast. Mold identification is discussed on the next slide. It's very different than the yeast identification. There are not magic biochemical panels or anything that you can use for molds. So what we commonly use to identify the molds is called the lactophenol cotton blue adhesive tape prep. This is the standard method that's used in most laboratories for mold identification. In the lactophenol cotton blue prep, you put some mounting media on a glass microscope slide, and then you use clear adhesive Scotch tape, and you touch the mold on the extending hyphal portions, get a good array of fungal hyphae onto the Scotch tape, and then press that into the lactophenol cotton blue on the microscope slide. The next step, of course, is to look under the microscope and visualize the type of hyphae that has been produced and if there's any conidia that has been produced. There is a picture of the lactophenol cotton blue prep on the slide, and you can see that it has kind of a lightish blue color background, and it helps stain the conidia and hyphae to make them more visible. If you're not able to identify the fungi using this phenotypic characteristics, you can use 16S RNA sequencing, and it is the best technique to use for problematic molds. However, the only laboratories capable of doing this are more your reference laboratories in mycology. On the next slide is pictured the safety precautions for use in a mycology laboratory. As you can see, the person is doing the work in a biosafety cabinet. This is a BSL-2 biosafety cabinet and will have HEPA filtration. This is very important when you're working on mold cultures. Mold cultures should never be worked on a benchtop. The conidia and hyphae can disseminate into the laboratory and be dangerous to laboratory personnel. With yeast, you do not have to maintain the same precautions. Yeast are not disseminated throughout the laboratory and can be worked on a benchtop. On the next slide, we start talking about the direct exams used to identify fungi directly from patient specimens. There are 4 techniques that are primarily used directly on patient specimens to look for yeast or mold. Gram stain, of course we use gram stain very often and is our most commonly method used in bacteriology, but gram stain is also very, very reliable for searching out yeast. It's not quite as good for molds, however. KOH preparations are a very old and standard technique to use to observe skin, hair, and nails for both yeast and hyphae. Calcofluor white stain. All specimen types can be stained and examined for both yeast and mold. And then the India ink, which is primarily used for sterile body fluids, particularly CSF, to look for Cryptococcus neoformans and gattii. On the next slide, we'll dissect each of these techniques a bit, and we'll start with the gram stain. The gram stain is very useful for the identification of yeast and not as quite as useful for identification of molds. Yeast cells stain very dark blue on the gram stain, so they are gram positive. You can also see the shape of the yeast, the size of the yeast, and you can also observe if there's any pseudohyphae present. Examination is usually required on oil immersion lens to be able to make out these characteristics. As you look on the bottom right-hand side, you can see what a mold looks like on a gram stain. It will sometimes pick up a bit of the safranin counter stain, but it does not stain very well, and mold can be really kind of hard to visualize on a gram stain, and is not a suggested technique. On the next slide, we break down the KOH prep, or potassium hydroxide prep. This is a very old method that's been used for decades in the mycology laboratories, but more and more labs are trying to phase out the KOH, as you'll see. In the KOH prep, how you do it is you put some KOH on a microscope slide, and then you put in your hair, skin, or nails, and they dissolve in the KOH. And then you put on a cover slip and push down, let it sit maybe for 10 or 15 minutes so the material can dissolve. And then you look at a 48x light microscope and try to observe for the presence of yeast and hyphae. If you look at the picture on this slide, you can see that it can be really difficult to differentiate the cell borders from the hyphae present on the slide. This is one of the reasons why this type of prep has kind of fallen into being not as popular as it used to be. They can be very, very difficult to interpret. On the next slide, it talks about the Calcofluor white prep. This is the Calcofluor white stain is how we've moved on from the KOH. The Calcofluor white is much easier to observe yeast or mold. The yeast and mold will bind with the Calcofluor white stain. You do have to use a fluorescent microscope to interpret this, and the sensitivity and specificity, however, is far better than the KOH, so it is certainly worth the effort to read on a fluorescent microscope. On the next slide is the India ink prep. This is also a decade-old preparation, but still can be useful, particularly if your medical center has a large number of HIV patients, because as you know, the Cryptococcus neoformans is one of the main infections of HIV individuals. You put 1 drop of black ink, literally it's like pen ink, so-called India ink, and it's placed, the 1 drop of India ink is placed on a microscope slide, and then you also put 1 drop of your CSF specimen, and then you observe it under a 40x microscope lens, a light microscope. It's a negative staining method because, as you can see in the picture on this slide, that the background is black and the yeast cell is visible, as is the clearing around the yeast cell, which is due to the polysaccharide capsule of Cryptococcus neoformans and Cryptococcus gattii. The specificity is certainly improved on this prep if you look for the presence of the budding of the yeast cell, and then you would less likely get confused with it being a white cell or some other kind of artifactual material. On the next slide, we talk about examination of fungi in fixed tissue. There's many types of stains that can be used in AP to visualize hyphae, but one of the most common ones is the methylamine silver stain, or GMS. Of course, on the GMS, yeast and hyphae stain gray to black. When you're looking at a GMS, you don't just look for the gray or black strands, of course, of hyphae or yeast cells, but it's important to also look at the presence for septations in the hyphae, the size of the hyphae. Is it large and broad or more narrow? Branching patterns in the hyphae. And we will look more of this later into the lecture, but it is important to generally observe characteristics of the yeast and of the mold on these stains. On the next slide is some pictures of PAS, or periodic acid shift stain. A PAS stain can also be used to visualize fungal hyphae in yeast. On this stain, the organisms will stain red against a green or blue background. On the next slide is the mucicarmine stain, or sometimes known as the mucin stain. This stain is specific for Cryptococcus neoformans and Cryptococcus gattii polysaccharide capsule. It will stain pink on this prep. This stain is virtually exclusive for Cryptococcus neoformans and can be used to help you identify C. neoformans and its close relative gattii in tissue when you are confused that it could be possibly another yeast species. On the next slide is the Hematoxylin and Eosin stain, of course known as the H&E. This is a great stain for looking at the cellularity of fungal infections, but GMS or PAS might be better for the actual visualization of the features of the fungi. But that being said, you can oftentimes visualize the hyphae in an H&E tissue. You can certainly see things like sulfur granules and such. So H&E is a very good starting point for the visualization of a fungal infection, and then you can move on to performing a PAS or GMS stain. On the next slide, we start talking about the dimorphic fungi. The dimorphic fungi, of course, are very important systemic pathogens with some very, very unique characteristics. On the next slide, we explain what does dimorphic mean. Well, a dimorphic means that there's two forms that exist for one fungus species, and depending upon the temperature and the conditions of the environment, you will express either the mycelial form or the yeast form. The mycelial form, of course, has hyphae and conidia. It is the free-living form found in nature, and we grow the mold at less than or equal to 30 degrees centigrade for a dimorphic fungus. The yeast or yeast-like forms are the parasitic phase of the mold, of the dimorphic organism, and they are found in the tissue or in laboratory conditions that we grow at greater than 35 degrees centigrade. At the bottom of the slide can show you some of the characteristics of the dimorphic mold, and this displays Histoplasma capsulatum. On the left-hand side, you can see when histoplasma is grown at 30 degrees, you get the mold form. You can see the hyphae and the large tuberculate macroconidia. On the right-hand side is the parasitic or yeast phase that occurs in the tissue. It is yeast cells 4 to 6 microns in size, and this would also be what would grow in the laboratory at 35 degrees. So same organism, however, due to its different conditions that you're placing the organism, you would either have a mold form or a yeast form. On the next slide, we talk about the dimorphic fungi. The ones that are most common are listed on this slide. You can see it includes Histoplasma capsulatum, Blastomyces dermatitidis, Coccidioides immitis, Paracoccidioides brasiliensis, Sporothrix schenckii, and Penicillium marneffii. On the next slide, we start talking about one of the most common of the dimorphic fungi, and that is Histoplasma capsulatum. This organism is found worldwide, but in the United States we think of the endemic area being the Ohio, Missouri, and Mississippi River valleys. But with migration of birds and winds and different atmospheric conditions, we are seeing the spread of histoplasma a little bit broader in the United States. But the classic endemic area remains the Mississippi River valleys. This organism is associated with bat guano or bat feces, and it's also associated with people who climb around in caves and are exposed to bat feces. And these people are known as spelunkers or cave explorers. It can also be associated with other types of bird droppings, primarily starlings, and also chickens are associated with Histoplasma capsulatum. On the next slide, we talk about the Histoplasmosis disease. 95% of infections with histoplasma are subclinical. You're climbing in caves or exposed to birds. You may or may not have a flu-like symptom or whatever, but it would be very subclinical or barely know that you're sick, but you would have a conversion of your serology. 5% come into very stark and realistic clinical infections. This can be progressive pulmonary, chronic systemic infection with dissemination to the reticuloendothelial system, including the bone marrow, and acute fulminating systemic disease if you have massive exposure to the organism in nature. This can, of course, be a fatal infection. Reactivation disease is also common and occurs in elderly and the immune suppressed. AIDS is a very good example, where exposure might have been in young childhood, but as an AIDS patient progresses in their disease, they would have recurrence of histoplasma. Bone marrow exam is very useful for looking for histoplasma. It is not useful for the diagnosis of many different kinds of infections, but become very useful in the diagnosis of Histoplasma capsulatum. One other thing that's unique about histoplasma infections is it can cause unique sites of dissemination. In histoplasma, one of them is the mouth and mucous membranes, as shown in this picture in the bottom right. On the next slide, we have pictures of what the yeast of Histoplasma capsulatum looks like in tissue. This organism prefers to be intracellular and inhabits the macrophage. The yeast are small, about 2 to 4 microns in size. They're very regular in size and shape, and you tend to be oval to round. Even though the name capsulatum implies that this organism might be encapsulated, Histoplasma capsulatum does not have a capsule, but it has a staining artifact. And if you look at the slide, particularly one on the bottom left-hand side, you can see as the yeast stained, it pulls away from the cell wall and looks as if it is encapsulated. And that's why it was originally named capsulatum. On the next slide, you can see how histoplasma will stain with a variety of different stains. It will stain with H&E, PAS, Gram stain, and also Wright stain. On the next slide, we talk about a rapid diagnostic test used for histoplasmosis, and that is detecting the antigen of histo in urine. It is a quantitative enzyme immunoassay. It can be done on a random urine specimen, and it's very useful for detecting disseminated infection and chronic pulmonary disease. The antigen of histoplasma is detectable in about 85% of these infections. It's good for diagnosis of immune-suppressed patients that do not produce a detectable antibody response. Antibody tests are available, but they've been very much replaced by the antigen detection in urine specimens. On the next slide, we start talking about what the organism looks like in fungal culture. Histoplasma capsulatum grows very slowly at 30 degrees centigrade. Sometimes it can take 2 to even up to 8 weeks to get a visible colony. The colony is white to brown and cottony on Saborod's inhibitory mold or your brain-heart infusion augurs. When you look under the microscope at a Histoplasma capsulatum mold, you will notice that it has tuberculate macroconidia, which are the identifying structure of histoplasma. These are large, about 8 to 16 microns in size, and there's also some small microconidia, about 2 to 4 microns, also produced. The microconidia are the infectious particle that's found in nature, and they are capable, due to their small size, of being breathed in and going deep into the lung to start the infection. Even though this colony, when you do a Scotch tape prep and look at it under the microscope, it has very characteristic macroconidia. Still, there are a few organisms that could be potentially confused with histoplasma. And when you're diagnosing someone with a dimorphic fungal infection that could take weeks and months of therapy, you want to make sure you're accurate. So laboratories should do a DNA probe confirmation or sequencing or some type of other test besides just phenotypic characteristics to identify Histoplasma capsulatum. You do not want any confusion if the person looking at the slide might be a bit inexperienced. The one organism that most literature will say looks most like histoplasma is a mold called Cepidonium species. It is a contaminant. It does grow more rapidly than histoplasma, but underneath the microscope, it does have some characteristics similar to Histoplasma capsulatum. On the next slide, you can see the appearance of the culture as it grows at 30 degrees centigrade. It is white and cottony. And there is a picture on the bottom that is a close view of what the tuberculate macroconidia look like. Tuberculations, of course, refer to the kind of spiny projections that come out from the large cells. And this is the structure, as I said, that we use for identification for histoplasma. The microconidia are also present. Remember, those are your infectious particle. And then you can see that there's hyaline septate hyphae also produced in this organism. On the next slide, you see the appearance in culture at 35 degrees centigrade. This is the yeast phase of the organism growing at this temperature. If you were to look at this microscopically, this yeast cell would be small and round to oval, consistent in size and shape of about 2 to 4 microns in size, and it has a narrow neck bud. On the next slide is the appearance of histo in tissue. Granulomas are usually produced and can be either caseating or non-caseating. The infection usually begins by breathing in the infectious particle, which is the microconidia, and setting up infection in the lung. So most primary Histoplasma capsulatum infections would be pulmonary infections. But this infection can disseminate to the organisms and the organs of the reticuloendothelial system, and it has very high dissemination to the bone marrow. So bone marrow is very often used as the primary specimen to look for disseminated histoplasma. In an examination of the bone marrow, you would look for the intracellular budding yeast cells present in the macrophages. On the next slide, I want you to be aware of look-alike organisms that can also appear in tissue specimens and could possibly be confused with histoplasma. Histoplasma is on the slide in the bottom right-hand side. And the two organisms most commonly confused with histoplasma could be Leishmania and Toxoplasma. I personally think Leishmania species is the primary one that could be confused with histoplasma. The cells are very similar in size, but the one outstanding characteristic of Leishmania that makes it different than histoplasma is the presence of the small round kinetoplast, which occurs next to the nucleus in the cell. So you will have a nucleus and then the very small round kinetoplast. Histoplasma does not have the kinetoplast structure. Toxoplasma could also be potentially confused with histoplasma, but I believe it's a little bit more banana-shaped, sickle-shaped, and is less likely to be confused with the round to oval histoplasma. On the next slide is presented the unusual variant of Histoplasma capsulatum known as variation Duboisii. Duboisii disease is primarily found in Central Africa. Unlike histoplasma, which is primarily found in the reticuloendothelial system, Duboisii variant is primarily an infection of the skin and bone. The 30-degree culture of Histoplasma duboisii is identical to that found in Histoplasma capsulatum. But the thing that is unique and different about duboisii is the yeast cells are 2 to 3 times larger than those found in capsulatum. If you go to the next slide, we will continue with a new fungus. This is Blastomyces dermatitidis. The epidemiology of Blastomyces is the Ohio and Mississippi River valleys. This is the endemic area in the United States. Unlike histo and some of the other fungal infections, there's no association with a specific animal or activity with Blastomyces. Most persons obtaining this infection have been in forested riverbanks, and it is thought this organism most likely grows in the forested areas due to the moisture and foliage in those areas. It's primarily a pulmonary infection, which may disseminate to the skin and bone. Pictured on this slide is a well-demarcated skin lesion that is the typical skin lesion produced by Blastomyces dermatitidis. If you were to scrape this skin lesion, you would see that it is full of yeast cells, which can help you, of course, in the identification of this infection. On the next slide, we talk about the culture of Blastomyces. It grows very well as a mold phase at 30 degrees centigrade. It takes 2 to 3 weeks to grow. It produces a fluffy white colony that's a bit prickly looking. If you look at the mold underneath the microscope looking at a lactophenol cotton blue prep, you will see pear-shaped conidia at the end of supporting hyphae. They look like somewhat like a lollipop extending out from the main hyphal shaft. There is a look-alike fungus named Chrysosporium species that you need to be careful in your identification of Blastomyces. And in doing this, it is always best not just to rely on the phenotypic characteristics or its look under the microscope. One would do a DNA probe or sequencing or something to assure that you have a Blastomyces dermatitidis in culture. On the next slide, we talk about what the culture looks like if it was at 37 degrees or the yeast phase of Blastomyces dermatitidis. It is a slow-growing yeast colony and can take up to 4 weeks to form a colony on an agar plate. The yeast cells are large. They're 8 to 20 microns in size, and it is unique in that it produces a very broad-based bud. And so that's one way you can remember this organism. Remember that Blastomyces is a broad-based budding yeast. I think that is shown well in the pictures in the bottom of this slide, where you can see a double contoured wall and very easily see that it is a broad-based bud between the mother and daughter cell. On the next slide is shown Blastomyces histopathology. It is normally a mixed pyogenic and granulomatous inflammation, which can be observed in tissue specimens, and you will of course be looking for the broad-based budding yeast cell, which is the pathogenic form found in tissue specimens. On the next slide, we talk about Coccidioides immitis. This fungal infection is endemic in the southwestern part of the United States. It can also be found in Mexico and South America. These are areas that are commonly known as the Sonoran Life Zone. They have a very warm climate and they have desert sands. Infection is from inhalation of fungal particles that are found in this sand. Coccidioides posadasii or posadasii is a genetically related to to Coccidioides immitis. These two species are very close relatives of one another, and for many, many years we only talked about Coccidioides immitis. But with taxonomic separation and good molecular techniques now available to us, they have found that there's actually two species of Coccidioides circulating around the world, and not just the one well-known immitis. These two species are primarily located in different endemic areas, but they produce the very same disease process, and they are morphologically indistinguishable in the microbiology laboratory. On the next slide, we talk about the infections of Coccidioidomycosis. 95% of the infections are asymptomatic or with limited symptoms. The remaining 5% are focal pulmonary, progressive pulmonary, or disseminated infections. Dissemination to the central nervous system is difficult to cure and has a very, very high fatality rate. In fact, it is the most difficult of all the fungal infections to cure. Higher dissemination to the central nervous system occurs in patients with defects in cell immunity, such as your HIV patients, darker-skinned ethnic groups, and in pregnancy. On the next slide, we talk about how the two species of Coccidioides looks like in a culture at 30 degrees centigrade. Unlike the other species of fungi that we have talked about, Coccidioides only requires 2 to 3 days to grow in culture. So it grows very rapidly as a waxy colony, and then it becomes more woolly with aerial mycelial. The progression from waxy to a full, mature mycelial culture usually takes 7 to 10 days. Because this organism can grow so rapidly, it can appear on regular microbiologic plates in your bacteriology laboratory. So you have to be very cautious in your bacteriology lab if you do see a mold colony to take it into the mycology area underneath the hood for observation. If you look under the microscope at a lactophenolcotton blue prep made of a cocci mold colony, you will see the septated hyphae with thick-walled barrel-shaped Arthroconidia. Please notice on the picture on this slide that there are dark, round-barreled Arthroconidia, and then a clear space, and then another barreled Arthroconidia. This is a so-called alternating barreled Arthroconidia. There is a dead cell in between the two live barrel Arthroconidia. What occurs is as the colony ages, these barreled Arthroconidia can break off from the hyphal strand, and they become the infectious particle in nature. This organism can be very infectious to laboratory personnel, so you have to use extreme caution when you're looking at any of these mold colonies. On the next slide, we continue looking at what Coccidia looks like, and also what Cocci could be confused with. Malbrachia species can look like C. immitis under the microscope. So this is the look-alike fungi that you primarily associate with Cocci. Because, once again, there is a look-alike fungi for this very, very important mycelial fungus, you want to make sure that you do a DNA probe or some type of sequencing to assure your identification of Coccidioides. On the next slide, we discuss a very important point with Cocci, and that is there is no yeast phase with Cocci, and this can be rather confusing for some. In the laboratory at 30 degrees and at 35 degrees, for both species of Cocci, we will get the barrel-shaped Arthroconidia. So no matter what temperature or conditions that we use in the laboratory, we will grow the mold phase of Cocci. Let's go to the next slide for the histopathology, and perhaps you can better understand the second phase, or what makes Cocci a dimorphic mold. On the Coccidioides histopathology, you will look at the slides and see there is not a yeast phase, but there's actually a thick-walled spherule that is produced in the tissue of specimens containing Cocci. These spherules are 10 to 80 microns in size, and inside you will see endospores. So this is the second phase of Cocci, and there are no yeast cells seen in tissue. Spherules can be seen in tissue in all stages of development, and sometimes they can be hard to recognize because they can be very small to very large, fragmented, and somewhat beat up in tissue. You will see granulomatous inflammation with caseation in these specimens with the spherules. On the next slide, you can see how the development of Cocci spherules occur in tissue. You inhale the barreled Arthroconidia that is found in nature. You inhale that conidia. The barreled Arthroconidia goes down into the lung, and it will continue to mature and produce a full mature spherule in tissue, which is very nicely displayed on the picture on the right. That is a very mature Cocci spherule with the endospores contained within. On the next slide, we can visualize other spherules that can also be found in tissue. But Cocci is not the only organism that can produce spherules in tissue. So it's important that you also know about an unusual disease in an organism known as Rhinosporidium seabori. This is actually an aquatic organism and is a parasite, but it can form spherules in tissue as well. But there are a few things that are strikingly different between the Cocci spherules and the spherules of Rhinosporidium. If you notice the spherules of Cocci on the left-hand side, they are usually, as I said before, about 20 to 80 microns in size. The spherules for Rhinosporidium are much larger. They're standardly over 80 microns in size. Also, the disease of Rhinosporidium is very different than the Cocci disease. Cocci is usually in your lung or disseminate throughout organs of the human, where Rhinosporidium is usually found as oral or nasal mass lesions. On the next slide is Paracoccidioides brasiliensis. This is also known as South American Blastomycosis, and the endemic area for this fungi is in South American countries of Brazil, Venezuela, and Colombia. People acquire infection from inhalation of the infectious particle in the soil, and most of the infections in this part of the world are in males. This is thought to possibly be due to estrogen inhibition of the mycelial phase of this organism, estrogen prevents the progression of the mycelial form of this organism to the yeast transformation. And so infections are far more commonly found in males. The disease presentation is pneumonia, dissemination infection to 2 or more organs, and extrapulmonary lesions that usually occur on the face and oral mucosa. On the next slide, you can see the laboratory presentation of Paracoccidioides. Cultures at 30 degrees for the mycelial phase are usually not done because this organism is so slow growing, and oftentimes you do not get characteristic structures produced on the mold phase. The culture at 37 degrees is slow growing, but the most characteristic thing about Paracoccidioides is the yeast that is produced. It's a large budding yeast, about 10 to 30 microns in size, and has multiple daughter buds, 2 to 10 microns in size. It is the only yeast that's going to produce more than 2 daughter buds, and it is so unique it has a specific name. It's known as the mariner's wheel yeast or the pilot's wheel yeast. If you look at the next slide, you can see the tissue examination of Paracoccidioides brasiliensis, and you can see the yeast cells are very, very large with very prominent daughter buds. You can also see a picture on the upper right-hand side of the nasal destruction that can occur with this yeast species. On the next slide, we'll talk about Sporotrichosis. This is caused by the organism Sporothrix schenckii. This is due to cutaneous inoculation of fungal spores penetrating injury with usually from thorns or some kind of a rose bush. The classic infection over time has been a patient being stuck with a rosebush thorn. The initial skin lesion can be with or without ulceration, and you get lymphocutaneous spread of the lesions as shown in this picture. Sporotrichosis can also, from the lymphatic, move into bone and become a systemic disease as well. There's also been reports of pulmonary and CNS infection, but they are rare and are less common than skin infections with Sporothrix schenckii. On the next slide, you can see what the ulcerative lesions look like for Sporothrix schenckii. In sporotrichosis, it can start as one or ulcerative lesion, and then it will start chaining up the lymphatics, causing infection of the lymph nodes and bone. On the next slide, we can see what the mold and yeast phase of Sporothrix schenckii looks like. The mold phase takes about 3 to 5 days to grow, and over time will turn brown to black. You can see what the lactophenol cotton blue prep looks like on the right hand of this slide. You get septate hyphae produced, very narrow, delicate looking hyphae, and then the spores are produced in a grouping known as the daisy wheel pattern. In the yeast phase, the yeast grows very well at 35 or 37 degrees, but they're small oval yeast cells. They're oval and elongated, about 2 to 5 microns in size, and this yeast cell is described as cigar bodies. On the next slide, you can see the histology of Sporothrix. It can be pyogenic to granulomatous. And one unique thing about Sporothrix infection is in tissue, it can be very, very hard to find the yeast cells in human tissue. Instead, you might visualize what is known as the asteroid body, or what is known as Splendor-Hopefully phenomenon. This is not specific just to Sporothrix. It can also be seen in other fungal infections such as the zygomycetes, Aspergillus, Blastomycosis in Canada, but is commonly associated with Sporothrix. This is an antigen-antibody phenomena which you can see in tissue. On the next slide is shown the lactophenol cotton blue prep of Sporothrix schenckii, and you can see the daisy-like spore arrangement of the hyphae and spores of Sporothrix. On the right-hand side, you can see the yeast cells that are produced. And notice that once again, they are cigar-shaped, and that is a unique feature of Sporothrix. There's also another picture of the asteroid body that can be produced. On the next slide, we talk about Penicillium morneffii. This is one of the unusual dimorphics. It produces a green colony with a red diffusible pigment on a Saborod's or inhibitory mold agar plate. And if you look over on the right-hand side, you can see what the mold form would look like under a lactophenol cotton blue prep. It is the standard colony morphology on the lactophenol cotton blue of Penicillium. But what makes this unusual is this is the only species of Penicillium that is dimorphic. It causes skin lesions in the tropics and can cause pneumonia in immune-suppressed individuals. On the next slide, you will see why Penicillium mornifii is termed a dimorphic. If you look at a tissue of a patient that has a skin infection with Penicillium mornifii, you will see that it has yeast-like cells in tissue. So this obeys all the rules of a true dimorphic. You have a mold at 30 degrees centigrade, and then if you look at the parasitic phase in the tissue specimen of the human, you will see the yeast-like cells.